Lipids might kind of get a bad rap, but they're really important biochemically. Your bodies rely on them for things like compartmentalization, so keeping things in places you want them to be and out of the places that you don't want them to be. Energy storage, we can have like our triacylglycerols, our tags that are going to be really great for kind of just storing energy in these blobs that won't soak up a bunch of water. Speaking of blobs, they're really good for like cushioning and insulation. They're also used in signaling, so we can have membrane lipids that can kind of play important signaling roles, and organization. So there are going to be proteins embedded in membranes based on like linkages to lipids. And so different types of lipids will play different roles. We have our triacylglycerols, so sometimes these are called triglycerides. These are our tags, those are for storage. We have phospholipids and cholesterol, which is an example of a sterol that are going to be used to make up membranes. And then we have modified lipids that are used for signaling. And then each of those is going to have a lot of variability within them. So let's dive in, talk about, first of all, some kind of nomenclature so we can be on the same terms about what some of these things are. And then we'll talk more about what they can do. Lipids can be a kind of wishy-washy term, but what they all have in common, lipids can be a kind of wishy-washy term, but what these all have in common is they have at least a large component of them is going to be hydrophobic. The classification of a lipid is a little bit wishy-washy, but what they all have in common is that they're largely hydrophobic. When we have hydrophobic things, well, then we've got to think about the hydrophobic effects. Basically, because water is polar and water wants to hang out with other water or with other hydrophilic things, other things that can kind of offer some charge or partial charge. If we think about a lipid, we're thinking about mainly hydrocarbon. Hydrocarbons are boring. Water doesn't like them. Therefore, water is going to maximize its interactions to things like other water molecules, and it minimizes its interactions with these lipids. In order to do so, it kind of squishes together those hydrophobic things, those things that it doesn't like, those things that can't offer the favorable binding opportunities. And in this way, you take less water out of commission and having to kind of surround each of those individual things if you push them together. Therefore, by pushing them together, you're freeing up water. When you free up water, you're increasing entropy. Entropy is good. This is good. Hydrophobic effect is therefore driven by this entropy of the release of water that you get because you're pushing the hydrophobic things together and not needing the water to surround each individual one by itself. So, hydrophobic effect, all about entropy. Now, though, once we have all those things pushed together, now, even though those hydrocarbons are pretty boring, because of the London dispersion forces, because of the fact that the electrons are moving all around randomly, some of them just happen to randomly move around into one side, and then they're squished next to another atom. Well, then the electrons in that atom are going to squish over too, and then you get a chain reaction, and you can have these molecular interactions form even between these hydrophobic things. And when these interactions form, well, then they can lead to kind of the solidness of some lipids. And if the interactions are less favorable, maybe there is shorter chains, maybe you have more kinks. It's harder for those hydrophobic parts to get close enough to have those kind of London dispersion forces be stronger. What's going to happen then is that you'll get something a little more fluidy. So you can get various properties just by changing hydrocarbon properties. A lot of lipids, however, although they are mostly hydrophobic or at least largely hydrophobic, they're going to actually be what we call amphiphilic or amphiphilic. This means that they have both hydrophobic and hydrophilic parts. So when we talk about phospholipids, or when we talk about detergents or soaps, detergent is just kind of like an artificial soap. What these have in common is that they have a water-loving part, a hydrophilic head, and then lipid tails. And then depending on kind of how bulky their tails are, so like in a phospholipid, you have two tails, and in a detergent, you only have one tail. This is going to influence then what happens when you stick these in water. And their heads are going to want to hang out with the water, or at least the water is going to want to hang out with them. And their tails are going to be pushed to the center. You can get various sorts of things like this. You can get membranes, you can get all sorts of stuff that make compartmentalization possible, that make cleaning up gunk possible. All of this is coming from lipids. So let's discuss lipid nomenclature a little so that we can then discuss the more fun stuff. Many lipids are derived from fatty acids, and so fatty acids are characterized by having a fatty part and an acid part. So basically that fatty part is going to be a long hydrocarbon chain. That's it. We call it its tail. These are typically 4 to 36 carbons long, and as you might imagine, that's going to be very hydrophobic. Doesn't have anything polar that water would like to hang out with. Then we have a carboxylic acid head. This head is going to basically allow for linking. So hopefully you covered in OChem how you can like join a carboxylic acid and an alcohol group to form an ester linkage. And basically this is what we're going to see is we're going to see that this head can be kind of like attached to different things. But that tail might sound really boring, but actually they can still have style. And so basically we can talk about these tails. They can have different lengths as well as different numbers of double bonds. So if we talk about like saturated versus unsaturated, with saturated, basically it's saturated with hydrogens. So all of those carbons are bound to their maximal number of hydrogens, meaning that we have all alkane and no alkene linkages. But they can also be unsaturated. When they're unsaturated, basically, instead of being bound down to as many hydrogens as possible, well, now they're sharing some of their electrons in a double bond. Now, this could be a cis double bond, in which case you kind of get a kink, or it can be a trans double bond, in which case you get more of a zigzag and not really changing the overall straightness of the chain. And so we'll talk about this more. But those are different terms that we could use. Is this like saturated or unsaturated? We can also talk about the degree of saturation. So if you only have one of those double bonds, we call it monosaturated. But if you have lots, we call it polyunsaturated. Both types can have different lengths. When it comes to referring to fatty acids, there are a variety of different strategies to go about naming them. Often what you hear is the common name. So you might hear things like palmitic acid or palmitate. That's our kind of default fatty acid. That's a saturated fatty acid with 16 carbons. And so you can see over here, we have this kind of notation that we can use for fatty acids, where we have a number of carbons followed by a colon, followed by the number of the double bonds. Like not where they are, but how many there are. So in the case of a saturated fatty acid, you'll have zeros on the right side of the colon. And in the case of palmitic acid or palmitate, we have 16 carbons. We see 16 before the colon. So the saturated fatty acids are easier because they're all just going to have zeros and we don't have to worry about locations of double bonds because they don't have any. But in the case of our unsaturated fatty acids, if we do have these double bonds, we need to refer to where those double bonds are as well as how many there are. So how many there are, that goes back to the number after the colon. And as to the position of them, we're often going to see them kind of written in the superscript when we're dealing with the delta notation. But there's also something called the omega notation. So let me just walk you through what these are real quick. I don't want you to worry too much about like the actual nomenclature, but I want you to be familiar with it if you see it. Basically, there's this delta notation and omega notation. Let's start with the omega. So starting at the end because, well, omega, that's Greek alphabet. That's the end of the alphabet. And so basically what we have here is we're starting from the end of the fatty acid when we count to find the position of the double bond. And we're going to be lazy with omega notation, and we're only going to look for the position of the first desaturation, the first double bonds. In this case, we go 1, 2, 3, and we see that this is an omega-3 fatty acid. You might have heard about like omega-3 and omega-6 fatty acids in like health stuff and things like that. Basically, omega-3 and omega-6, our bodies can't make those fatty acids from scratch. The way that our bodies make unsaturated fatty acids is they actually use an enzyme, a desaturase, to actually introduce those double bonds from the default saturated fatty acid that they make. but we don't have enzymes capable of putting in omega-3 or omega-6 double bonds. And so what this means is that if we want omega-3 or omega-6 fatty acids, we got to eat them from our diet, and then we can modify the ones that we get through our diet. But we need those from our diet originally in order to have them. That was omega notation. So we count from the end to end, and we are going to just count the position of the first one. In the case of our delta notation, well, here we're a little more systematic. We're going to start at the beginning. We're going to start at kind of the fatty acid end, the end with our carboxylate. And we're going to count from here. So starting with the carbonyl carbon, we have our 1, 2, 3, 4, 5, 6, 7, 8, 9. So in this case, we have 9. We have our position at 9. And so we're going to indicate this as this delta superscript 9. And this would be our cis-9 octadecanoic acid. So it's cis because this is a cis double bond. Octadecanoic, it has 18 carbons. So decanoic, that's like 10. Octa 8, so this is 18. And we could give this a notation, this one a delta notation as well. And so in this case, we had one double bond, so we had 18-1. Here we have 5 double bonds and we have 20 carbons, so that's why you get the 25. And then delta, and those are just the position of those double bonds. So there are a lot of different possible fatty acids, but there are some common saturation patterns. You don't need to worry about this. Just know that there are a lot of fatty acids, but they're not as many as there could be in a way. We can refer to them in general terms as either saturated, so no double bonds, or unsaturated, so they have some double bonds. If they just have one, it's a monounsaturated, so a MUFA. And if it's got a lot, then it's polyunsaturated, so a PUFA. Looking back at our table, we also see some trends that as the length of the fatty acid gets longer, well, what's going to happen is that there are going to be more interactions that can happen between different copies of the fatty acid, more of those dispersion forces, those van der Waals. So what's going to happen is they're going to stick to each other more, and so you're going to get an increase in your melting point as you go longer. But if you introduce double bonds, that's going to prevent the tails from getting close together and forming those interactions. And so when you're adding double bonds, when you're adding desaturations, you're going to be decreasing the melting point. You're going to be making it easier for it to melt. And so you're going to see that as you introduce more and more of these double bonds, the melting point is going to get lower and lower. So you're going to end up with going from something that maybe was a solid at room temperature to something that was a liquid at room temperature. And this could be the difference, say, between your fats and your waxes and stuff and your oils. So longer chain, higher melting point, more saturation, higher melting point. And at the room temperature, you're going to see those saturated fatty acids are going to have be solid and have a waxy consistency, whereas the unsaturated fatty acids are just going to be oily liquids. And how close together those tails can be is going to depend on the degrees of saturation, as well as whether how kinky they are. So are they trans double bonds or are they cis double bonds? Say you want a more solid form of something that is an oil at room temperature. Well, you can imagine that one of the ways that you could do this would be to hydrogenate it. So basically to remove those double bonds, allow there to be more of those closer packing, allow there to be more of those weak interactions that are going to collectively make for a higher melting point. So you can say, take a vegetable oil or take a fish oil that has that omega-3 that you need, and then convert it into a more solid form. Then you can do things like make your cookies with. Scientists can do that, or labs can do that. Manufacturers can do that. They can basically use hydrogen, introduce hydrogen in order to reduce those double bonds to single bonds. Another benefit of this is that basically those double bonds can undergo oxidative cleavage. Over time, this can cause lipid-rich food to spoil when those double bonds get cleaved. So if you could basically turn them into single bonds, well, then you wouldn't have that problem. You could improve your shelf life. What could go wrong? Well, basically, you wouldn't want to change all of those double bonds usually to single bonds, because then you would have something that was just like a solid rock. It wouldn't be very useful for your cooking. You need something that's kind of a little more It's kind of solidy, but more slippery. I mean, you can actually like work with it. And so typically the hydrogenation isn't done like fully, instead it's only done partially. And well, when this happens, it's going to have the effect of accidentally like having this byproduct where it's going to convert some cis double bonds to trans double bonds. All of the natural fatty acids have cis double bonds. So this is basically introducing these unnatural trans double bonds. And it turns out that these trans bonds, so these kind of the more zigzaggy ones, not the ones that actually like kink you, not only do they alter the properties of the lipids, but it also has problems health-wise. Natural fats and oils are a mixture of simple and mixed, saturated and unsaturated tags. So different, like plants will have differences versus animals and various things like this. Now that we're familiar with fatty acids, let's go ahead and link them up to form some more interesting things. Let's talk about membrane lipids as well as storage lipids. And so I guess storage lipids aren't that interesting. They're kind of just blobs, but they're really important blobs. Thinking back to our hydrophobic effect and how these hydrophobic things are going to be forced together because water is going to exclude them. Well, if you want to store something long term and you want to like have a nice condensed storage form, this is actually really, really great. We wouldn't want something like sugar that was going to be soaking up a bunch of water. So remember how we talked about how we can make like agarose gels and things by taking advantage of the fact that sugars are going to attract water and you get this kind of gel mesh? Well, that would be like a huge water weight that you're carrying around in your body. But lipids are going to allow us to have this compact form of energy. We don't have that benefit we had of glycogen, so like of our sugar storage. We don't have that benefit where we can kind of easily chew off little pieces. So remember, glycogen had all that branching that made it really easy to chew off pieces and get energy. So our fats are going to be harder to mobilize. But once we do mobilize them, then they're going to actually give us more bang for our buck than our sugars will. And this has to do with, we'll talk much more about this, but this has to do with the fact that these fats, they're going to be in a more reduced form. They're going to have more potential to be oxidized. And when we oxidize them, what we can do is we can reduce electron carriers. So what's going to happen is that when you break down fatty acids, which is done mostly in a process called beta oxidizing, you're oxidizing this fatty acid. And in the process, what you're doing is you are reducing NAD to NADH and FAD to FADH2. And then your bodies can go and use that in the electron transport chain, oxidative phosphorylation, make some ATP, booyah. And so if you're starting from a carb, well, now you already have a bunch of OHs and stuff. You've already done a bunch of the oxidation. And so you're going to be kind of starting lower down than you would if you were to start with a pure like hydrocarbon. So fats are really great for storage because of the fact that the water isn't going to be like wanting to hang out with them. And because of the fact that they have more potential to be oxidized, that is, they are in a more reduced form than your sugars. So they're good for that long-term storage. And so we want to basically neutralize even just that carboxylic acid head. And so what we're kind of want to have is something like a triacylglycerol. So triacylglycerol, you can also see this called like triglycerides. These are often abbreviated as tags. In these cases, basically what happens is you have this scaffold molecule, this glycerol. So glycerol is this simple little molecule. We have these 3 OH groups hanging off of our 1, 2, 3 carbons. So 3 carbons, 3 OHs, 3 places to link on. And we can hook up one of these OHs to our carboxylic acid. And then therefore we're going to form this like ester linkage. And the triacyl, so the acyl part, that refers to kind of like a carbonyl next to a carbon next to something. And so basically what's going to happen is that this part is like a generic acyl. And in the case of our triacylglycerols, we have three acyl linkages. So we have like an acyl here, an acyl here, and an acyl here. And so these would be acyl glycerols. And we also can refer to these as ester bonds. And the acyl is just referring to kind of the fact that we have this carbonyl carbon next to something. So acyl is this part, and then the glycerol. It's just kind of a weird naming thing that you don't need to worry about too much, but that's where the name comes from. In a triacylglycerol, like the name suggests, you've got these three fatty acids. These fatty acids can be different forms, so you can have different lengths, you can have different degrees of saturations. If you want something that's going to be super duper globby, you wouldn't want much in the way of unsaturation, not much double bonds. Also, when you have the double bonds, you're already kind of oxidizing it part of the way, so you're losing some of the potential to get energy later. The reason why those triacylglycerols were kind of sitting around as blobs is because they're just like really unreactive. They're basically mostly just hydrocarbons. They are really not non-polar, just like fully almost. But what if we have something that is partly hydrophilic and partly hydrophobic? Well, in this case, instead of just forming like a clump of nonpolar hydrophobic stuff, now if we were to stick those into water, we can do things like form membranes. And so we can have, say, a phospholipid, which has a hydrophilic head and hydrophobic tails. We can put that into water and get a membrane. And so let's talk about our membrane lipids. So when we talk about membrane lipids, so remember, what do they have to have? They've got to have that hydrophilic head and those hydrophobic tails. And so when we have something like this with hydrophilic and hydrophobic parts, we call it amphiphilic. So amphi for both, and then philic for loving. We can also call these amphipathic. And the key ones that we're going to talk about are phospholipids, which have two fatty acids joined to glycerol or sphingosine. So in the case of our triacylglycerides, we had three fatty acids joined, three of those tails. And in the case of our phospholipids, we're just going to have two tails. But then basically one of the other carbon in the backbone is going to be used to form a bond to something that's polar. So it can be polar, it can be charged. And so that's why we kind of have two tails, because, well, one of the glycerols is being used, one of the alcohol groups from the glycerol is being used to make this connection to our hydrophilic head. Additionally, we can have phospholipids that instead of having a glycerol backbone, they have this thing called sphingosine, and so we'll talk about it in a minute. Now, these phospholipids are going to be interspersed with other things like glycolipids, where basically they have a simple sugar or a complex oligosaccharide. So one of those like glycan things at their polar ends. So basically it's just sugar on a lipid. So you can get sugar sticking out of your membrane, like things like those ABO blood types sugars that determine that. It's like we talked about. Now, there are also sterols. So our key one is cholesterol. And these have a rigid system of 4 fused hydrocarbon rings. And then we'll go into how the structure of that cholesterol is going to help keep our membranes fluid. So basically, this is a little intimidating of a chart. So hopefully I can kind of steer you towards what we're going to focus on. So let's start with those phosphoglycerides, our glycerophospholipids. And remember that those are going to have our glycerol backbone and attached to 2 fatty acids. Through 2 of those positions, 2 of those OHs, and then the third position is going to have a head group that is going to be linked through a phosphate. And so the 3 key ones that you need to know, you don't need to memorize their structure, but you should know the names: ethanolamine, choline, and serine, in which case we would call it phosphatidylethanolamine, phosphatidylcholine, or phosphatidylserine. As for those tail positions, well, just as we saw before, these can kind of have like different lengths and different degrees of saturation. Thankfully for us, things are kind of simpler. Instead of all those different combinations, you typically have in the first position, like a C16 or a C18 saturated fatty acid. And then at that C2 position, so that second position in our glycerol, you typically have a C18 or C20 unsaturated fatty acid. So remember, unsaturated means it has those double bonds, it has those kinks that are going to make it so that the chains can't get as close together. And this is going to make it so that our membranes have some fluidity, since we kind of have one of those straight chains and one of those kinky ones. And then in the third position, that's where we're going to have our highly polar or charged head. that's going to be attached to the third OH position in our glycerol through this like phosphodiester linkage. Those head group constituents, remember that's the part that can vary. And so we have things like our phosphatidylethanolamine, phosphatidylcholine, and phosphatidylserine. But then you can get all sorts of other derivatives. In terms of our other type of phospholipid, our sphingolipids, basically here for their backbone, it's also one of their tails. So it's a little confusing, but sphingolipid, instead of having a glycerol backbone and two tails, two fatty acid tails, it has a single fatty acid tail, but it also has a tail that's coming from the sphingosine. So the sphingosine itself, it kind of has this L-shaped. And so it takes the position of that glycerol, where you have that one position is attached to your head group. Then you have one position that's going to be attached to a fatty acid. And then the other position, it's kind of just attached to the hydrophobic chain of sphingosine. So this molecule sphingosine is what our backbone / tail is for one of them. And then we have, instead of having an ester linkage, we have an amide linkage because we have it attached through a nitrogen. There are different types of things that then can serve as our head groups, just as we saw before. They have important roles in your body that we're not going to be able to get into. But as the myelin might suggest, these are going to be really important for like myelination and things in your neural tissue. They're also important for cell signaling and cell surface recognition. Okay, we do need to, though, get into our sterols. So remember, our main sterol we're going to be talking about is our cholesterol. Well, what all sterols have in common is they kind of have this core structure of 4 fused carbon rings. So this like A, B, C, D. This is showing you the structure of cholesterol, but there could be other sterols that still have this structure. In addition to this like core, which is almost planar and fairly rigid, it also has this like alkyl side chain, as well as a polar head group that's going to allow it to kind of have a little polar part that can stick out into and hang out with the hydrophilic heads of those other guys in the water, but then kind of like hide the rest of it in with all the nonpolar tails of those others. Additionally, sterols can form as precursors for hormones, testosterone, cortisol, prednisone, aldosterone. All of these are basically steroids, which are just oxidized derivatives of sterols. So you can see that they still have those like 4-ring structures, but these are going to have more oxidation to them, and they're not typically going to have like that long nonpolar tail. And these can form important things like hormones, various cool stuff. Speaking of various cool stuff, there's a lot of types of lipids that we didn't get into and that we're not going to get into, but your book goes into if you're interested. Waxes are kind of just esters of long chain fatty acids, allow you to have things like bunny bees do cool stuff. And similarly to other lipids, they're going to repel water. In the case of a wax, it's kind of going to have this, waxy consistency. So they have, as Anna mentioned, they're going to have like a higher melting point to keep them that solid, that really hard solid. Okay, but let's talk about things that are less solid. Like we want our membranes to be solid. Instead, we want our membranes to be fluid, and we want our membranes to be dynamic. And we need to talk about what's going to happen when our amphiphilic lipids meet water. Not just that super hydrophobic wax that's just going to repel the water, but our amphiphilic lipid, which has that hydrophilic part, which will hang out with the water, and that hydrophobic part, which will be excluded by the water. What would happen if you put that into water? Well, it depends on the shape of that amphiphilic lipid. If you have something like a soap or something like a detergent, where basically what you have is you just have a single tail and a head, where the cross section of that head is going to be greater than that of the side chain, you kind of have like a coney thing. If you stick that into water, what's going to happen is it's going to form this thing called a micelle, where basically all those nonpolar parts are kind of just going to be in the center, and there's no room for anything else around it. So there's no liquid inside of here. We have the single layer, and it's just like this blob of gunk inside, and then this hydrophilic outside. You can actually take advantage of micelles in the lab to do things like purify out membrane proteins because they form that nice kind of little lipidy bubble thing. But that wouldn't be very good for our cells if we couldn't have anything liquidy inside of them. And so instead, we need something that instead of having that shape like a cone, we have a shape that's more like a cylinder. And where did we see a shape that was more like a cylinder? Well, with our phospholipids, because remember, we've got those two tails there instead of just a single tail. So we have this more cylindrical shape. And now it's not going to be able to form that micelle because it can't kind of compact that way. Instead, in sticking into water, what's going to happen is it's going to form a bilayer. So it's going to form this kind of like sandwich where those hydrophobic tails are going to be in the center and the hydrophilic heads are going to be on the outside. And if you have enough of this that it like folds up on itself, well, now you can get some sort of like vesicle where you actually have liquid inside. And then you have this bilayer membrane. And you can imagine that, so this would be also called like, these vesicles are sometimes called like liposomes. You have these like small ones, but now imagine that you have a lot of liquid in there. You can get these really, really big compartments. You can have cells. And then inside of cells, you have all sorts of different compartments. So inside of our cells, there's actually like a huge interconnected network of membrane-bound compartments. So we have those like bigger things like our mitochondria, our nucleus, our endoplasmic reticulum, and our Golgi apparatus, which is where the kind of like some proteins are processed and stuff to be made to be secreted or otherwise modified. But then we have smaller compartments like our lysosomes, which break things down, and endosomes, secretory vesicles. We have all sorts of these membrane-bound things that are kind of all interconnected through this dynamic endomembrane system. And different membranes within that system actually have different compositions. So we saw all that variability we could have with our fatty acids and with our phospholipids, with our glycolipids, all those various things. And they're going to differ between different compartments in the cell, between different membrane-bound compartments, and even between the two leaflets of the bilayer. So when we talk about that bilayer, we talk about like the outer leaflet, the one that's actually facing the exterior, and then the inner leaflet, the one that's facing the interior. We call those leaflets, and those can have different compositions of lipids. So we've been talking about lipids, but I've got to tell you, membranes are not just lipids. In fact, about half of the mass of some of our membranes comes from proteins. And so because lipids are much smaller, there's about 15 times more lipid molecules than protein ones in your membrane. And so really thinking in terms of lipids is going to miss a lot of the picture. You'll also, membrane proteins are super duper important. About a quarter of our protein coding genes are thought to be for membrane proteins, and a lot of drug targets. So when we talk about like membrane proteins, we'll talk about various types of proteins. They can either be on the surface or go through the membrane. They can be transporting things. They can be signaling things, all sorts of really cool stuff. Key thing to know now is that basically the proteins are a really important part of the membrane. And you often hear of this like fluid mosaic model where basically individual lipid and protein units are kind of embedded in a membrane and moving all around. You might hear about stuff like rafts where there are regions that are kind of stiffer. There are various proteins anchored in various regions. The lipids, they can kind of diffuse laterally so they can go side to side. But they need help actually crossing the barrier because like in flipping to the other side, because we'll remember that means putting their hydrophilic heads in the hydrophobic surface. And so some of the membrane proteins we'll talk about actually help with that. And so there are flip bases and flop bases and scramblases, and you don't need to know their actual names, but those are fun names. And so those are going to help the lipids kind of get where they need to go if they need to switch sides. And so this is a way that your cells can kind of move things from the inside to the outside on the heads of lipids, as well as kind of change the composition of their membranes so that different cellular compartments can kind of specialize. The fluidity of that membrane, the ability for things to actually have that fluid mosaic model is going to be dependent on the membrane, not just kind of being stuck in place. And so if we had membranes that were made up out of that like triacylglyceride, well, not only would it not have that hydrophilic head to do things, but if you had those three tails that were coming really close together, if you had a fully saturated membrane, what's going to happen is that you're going to have that tight packing. You're not going to be able to do that diffusing. You're not going to be able to move things around easily. You're just going to have like a solid clump, which would not be good. And so we want something that's going to be more fluid. And so that's why we have things like we have our unsaturated fatty acids in our membranes. We introduce those kinks. So remember, you typically have one saturated and one unsaturated fatty acid per phospholipid, so that we can get those kinks, we can get some of that fluidity. And we also have that sterol. We're going to have that cholesterol that we'll talk more about in a second, because it's going to play a key role in dictating the fluidity. What also dictates the fluidity, though, is temperature. So even if you have that beef fat, if you get it hot enough, you can melt it. Similarly, instead of talking about like a melting temperature, when we talk about our membranes, which are this like heterogeneous mixture, so basically you have a bunch of different stuff in there, we talk about this like transition temperature, where basically below that transition temperature, things are kind of more like solidy, they're kind of like stuck in place. But then above that, you get this more disordered. You get things moving around, because remember, what is temperature? Temperature is just like heat, it's energy. It's the amount of like energy that they have. And so if they have more energy, they're able to move around more, they're going to have more fluid composition, more disorder. And so that's going to, if you have an energy that's enough for that, that's what you're going to see. But below that, you're going to see kings kind of just like stuck in place more. And that transition point, we call the transition temperature. Now we call it a temperature, but really it's not like everything goes from one thing to the one to the other like instantaneously. So instead it's more of a kind of like range. But what happens is if you just had phospholipids, you would see that you would have this kind of sharp inflection point at your TM. And basically what this would mean was that you didn't have much of that wiggle room. And so if you were to go slightly above the TM, well, now you would have everything kind of be all liquidy, all like oily kind of. But if you were to go below it, you would have things be all solid really quickly. And so you wouldn't have much of a buffering area. You could quickly get yourself in trouble, have your cells all melty, or have your cells all like stuck in place. And so we need to add this sort of buffer. And the buffer that we're going to use is going to be our cholesterol. So not a pH buffer, we're not talking about pH here, but just a buffer in terms of kind of allowing ourselves a little bit of safety space so that we don't have that sharp transition. In this case, what we're going to use is our cholesterol. So remember, cholesterol was an example of a sterol. It's got those 4 rings, and it's got the hydrocarbon tail. And then it had that OH on the end. It's that one little polar part so that it could hang out with those polar head groups. But then what's going to happen is that nonpolar part is going to be interacting with the phospholipid tail next to it. And because this ring system is going to be less flexible, it's kind of going to stiffen up this region. Then you're still going to, at the end, kind of get a more fluid region. But you can think of this cholesterol as kind of like interacting with these things. If you're at high temperatures, when all the stuff around it is able to be really flexible, when all of this region of the phospholipids kind of like moving all around, well, here the cholesterol, its ring system can't move around easily. And so this is going to help stiffen things up when you're at those high temperatures. And it's going to reduce the fluidity at those high temperatures. But what happens at low temperatures? I mean, like in order for a buffer to be, well, a buffer, we need it to be helpful at low temperatures too. And at low temperatures, we would need it to increase the fluidity. And it does this because what it's going to be doing is it's basically interrupting the packing of these phospholipids next to one another. So at lower temperatures, it's kind of preventing things from forming those interactions that would hold things in place. And instead, it's kind of going to be making it take less energy to make things fluid. But then when things are starting to get all fluid, well, now that cholesterol isn't getting all fluid. And so now it's going to be harder to get it even more fluid. It's going to be harder to kind of make it oily. So in that way, cholesterol is able to act as a sort of fluidity buffer. So we have our triacylglycerols. Those are going to be used for our storage. Those have those glycerol backbone and those 3 fatty acid tails. Then we had our phospholipids and cholesterol that were making up the membranes. We had our glycerophospholipids and our sphingospholipids with the glycerol or the sphingosine backbone, and those like 2 tails, and the hydrophilic head, and a lot of variability within each type. But all of these are going to allow for things like compartmentalization, having those phospholipids and glycolipids that are making up our membranes with the help of sterol, kind of keeping things fluid. Those triacylglycerols forming those like energy storage and cushioning and insulation. And then we had our signaling as well as like organization, having sugar chains and having proteins attached to the membrane proteins. So all sorts of really, really cool stuff about lipids, all sorts of stuff we didn't even get to, but I think that was enough for today. Hope that helped.